All right. Well, thank you for inviting me to talk about a little bit of the project. I've been working at Cornell for 29 going on 30 years, and there's been a lot of changes. Computers were just starting to be used when I came, and there was no such thing as molecular genetics. And my point is that if you are not able to change with the times and learn new technologies and acquire new knowledge, you'll be left behind very quickly in plant breeding especially. So what I'm going to talk about today is a little bit about our marker-assisted selection program and a little bit about some of the traits that we're working on, and then a little bit about where I think plant breeding is going in terms of some of the new techniques. This department has been active for 100 years this year. The small grains breeding program was started at the same time that the department was started, and so we're 100 years old also, even though I don't look like it. The small grains project objectives don't really change very much. These are pretty similar to the objectives I started out with years ago. And the reason is because plant breeders have very common objectives that they need to address when they're doing plant breeding research. We need to understand the inheritance, chromosomal location, and expression of genes that control the traits we're interested in. My particular interest is breeding strategies. Now, some plant breeders will have the objective of understanding the genetics and the manipulation of pathology traits or physiological traits or insect resistance, but I'm interested in breeding strategies. All plant breeders use different kinds of breeding strategies, but not everybody tries to develop new strategies. And lastly, most plant breeders are interested in developing superior germplasm and varieties, or varieties. And by superior, I mean that they're improved for one of the many characteristics that are required to develop something that you can call improved. I'm going to start with a couple of projects that I've worked on for the past 7 years. The first project was a 4-year project that involved collaboration with 10 other labs around the country. It was funded by the Initiative for the Future of Agriculture and Food Systems. And it was called Bringing Genomics to the Wheat Field. It was started in October of 2001, and it involved all the different kinds of wheat that are grown in the United States. That project has been completed, and we're now working on a project called Applied Wheat Genomics. This is a project funded by the USDA. Again, it's a 4-year project. This time it involves 25 different plant breeders around the country. Again, all different market classes, all different kinds of wheat, and the regions are shown here. This is the eastern region, that's where we are, north central, central, and western regions. And each region has a regional coordinator. In addition to developing new markers and doing marker-assisted selection, we also coordinate education and outreach activities and attend annual symposia and planning meetings. This is what our website looks like. The website is very comprehensive. It includes protocols that we use, information about the project, details about the project, and information on the different mapping populations that we're using to develop new markers. It also has information about the germplasms and varieties that we release as a result of this project. And down here, you can't see it at the bottom, is a list of publications and presentations. This is just one example of a web page that shows the characteristics of the mapping populations that we're using. Highlighted here on the right side is the one that we use in our lab called Cayuga and crossed to Caledonia. And you can see that it lists the number of lines in the population how it was made, different traits we're interested in, the contact, and the market class, and also the pedigrees of the parents. So if you're interested in mapping traits in wheat, you can go to this web page and pick any of these populations to learn about what traits are segregating. Extremely useful. We also characterize these populations for the traits and the genes, the major genes that they have, and you can tell which ones are segregating. For example, here we know gluten strength is segregating because one parent has the number 7 high molecular weight glutenin, and the other one has 7 plus 9. So we know that that's segregating in this population. And they're also characterized for disease resistance, aluminum tolerance, vernalization, height, and so forth. This figure just shows you some of the traits that we work on, as well as a conventional, I would almost say old-fashioned approach to marker-assisted selection. Some of the traits include quality traits listed here and disease and insect resistance listed here. The ones that we've worked on in our lab are starch quality, pre-harvest sprouting, and hardness, actually softness, technically speaking. The disease resistances that we're interested in are Fusarium head blight, eye spot, barley yellow dwarf virus, and wheat spindle streak mosaic virus. Most of the other diseases and insects are not a problem in this region. Now, in a typical backcross breeding scheme, which I'm sure you've studied already in this class, you have a source parent and a recurrent parent. The source parent has the trait you're interested in, but the recurrent parent lacks that trait. So the idea is to make the cross, and in a backcross 1, select in the backcross 1 segregating population for the heterozygous plants and make a second backcross. Of course, if you can't see which ones in the backcross 1 are containing the gene for the trait, you have to self and select in the self population. With markers, with codominant markers, you can easily see which individuals in the backcross population have the allele that you're interested in. And I'll show you some examples of this as we go through some of the slides. New York is in the soft white winter wheat growing region. That means that our kernels are white instead of red. They're soft instead of hard. The hard wheats are primarily used for bread-type products. The soft wheats are more all-purpose. They're used for many different kinds of products, including cookies, cakes, crackers, breadings, breakfast foods, and many others. The white wheats, soft white wheats tend to be lower protein than the hard wheats, and that's because you don't need as much protein to produce those kinds of products. Oops. The kernels are white, and that's a special characteristic that is very useful for whole grain products. When you try to toast products like Triscuits that are made from red wheat, they come out looking kind of a dirty brown or almost black. Whereas if you use white wheat, the bran toasts up to a nice golden brown, which consumers like. Winter wheats are planted in the fall, and they go through the entire winter, and in cold regions they actually go dormant in the winter, and they can tolerate extreme cold and heaving and freezing conditions. So they're very tolerant of those adverse conditions. The spring wheats are planted in the spring, typically April or May, and as a result, they tend to be lower yielding. And the reason is because they haven't had the advantage of growing in the fall and getting an early start in the spring when farmers still aren't in the field. So they have a longer growing season, and they're able to be more productive. The winter wheats are able to be more productive. Now, I wanted to say a few words about quality. I mentioned that we have hard wheats and soft wheats, and that the hard wheats tend to have higher grain protein. They tend to have greater gluten strength, and there's a complex locus on chromosome 5 that conditions hardness of the kernel. The gluten strength is partly controlled by these high and low molecular weight glutenins. They're a kind of seed protein. I won't go into all the detail, but for us, the high molecular weight glutenins seem to have the most influence on viscosity. And of course, viscosity is important because when you're making bread and you knead the dough and the yeast is growing in there, the proteins have to trap the CO2 from the yeast, and that gives it its nice airy texture that we all like in a nice bread, nice type of bread. So typically we have information about each of these different kinds of high molecular weight glutenins and which ones are desirable and which ones are less desirable. So we can use that, either a page gel to select those or markers for those genes. The high molecular weight glutenins are located on all of the three wheat genomes, and chromosome 1A has essentially two different kinds. The 1B chromosome has 5 different kinds, and most of ours have the 7 and 9, which means that they are not very strong gluten. The D chromosome, D1 chromosome, has 4 different kinds, and most of ours are sort of intermediate here with the 2+12 types. So that just gives you some idea about how we can alter gluten strength using molecular markers. Yes?
I don't understand what like 2+12 means.
It's a very simple, just a designation that was developed many years ago. When they looked at these page gels, they just numbered them 1 through however many there were. So it really doesn't have any significance as far as the functionality. Yeah. So it's just like calling Bob or Jack or Linda or something like that. I won't have a lot of time to go into detail on some of these characteristics, but I just want to give you an idea of the kinds of markers that can be used for wheat. Now this happens to be an interesting marker, an interesting trait that has markers for a complex locus on chromosome 5 that consists of 2 genes that code for a protein called purulindolin. It also codes for a protein called grain softness protein. These proteins work together to make a kernel soft. It's called the hardness locus, but these functional genes actually produce a soft kernel. If any of them are non-functional, then you have a hard kernel. Now, a typical hard wheat or semi-hard wheat is shown here, a normal control. And what we would like to do for our region is to make the wheat softer. And the reason we want to make it softer is because softness makes it easier to produce a high-quality, uniform product. When we have a few hard kernels in the processing, then you sometimes get incomplete cooking of those kernels. If we add from a wild wheat relative a similar locus to our cultivated wheat, it makes it softer. It's called double pin A, double purulin. If you have a defective pin A, the kernel gets harder. If you have a null for all 3 of these genes, it gets even harder. If you have a null for all 3 genes plus the a modified 5D chromosome, it's even harder. So you can modify the hardness by adding or subtracting these genes to the cultivated wheat. This gives you an example of what one of the markers looks like for the purulindolins. This is a marker for purulindolin A. This is a marker for purulindolin B. And remember I mentioned that if either one of these is defective, then you get a soft kernel. So any of these that are lacking a band here are hard, and you can see here that these are hard. If you look over here, the same situation. Anyone that's lacking that band is hard. You only get a soft when you see both of them present, and that's only present here in both of them. So that is what you need to have to get a soft kernel. Now this is a codominant marker for the puroindolin B. In this case, you have to digest in order to be able to see the codominant marker. You digest it with a restriction enzyme. That's how you can see the soft allele. So there's a bit of a complication there, but this is called a CAPS marker, cleaved amplified polymorphism. Now I'll say a few words about waxy endosperm. Waxy endosperm is endosperm that has a high proportion of branched starch. In typical wheat kernels, you have a mixture of straight chain and branch starch molecules. But when you have a defective gene for the granule-bound starch synthase, then that results in a higher proportion of branched starch. And in maize and in rice, there's one gene for this, but because we're working with a hexaploid that has 3 genomes, we have 3 genes. So that means to get a high proportion of branched starch, we need a defective gene at all 3 loci. Now we can detect small differences in the amount of branched starch if we have a defective gene at any one of those 3. But when we have a defective gene at all 3 loci, then we have a very high percentage of branched starch. And this is the 7D waxy locus, this is the 4A waxy locus, and over here is the 7A waxy locus. So we have a different marker for each one of those 3 genes. And by using the markers, it's much easier to see when you have a 2 or a 3 gene segregate than if you are just trying to measure the amount of branched starch. I've been working on a trait that is important for this region ever since I came to Cornell. White wheats tend to have lower seed dormancy than red wheats. The red kernel color seems to be pleiotropic for higher seed dormancy. That means that it affects both seed color and seed dormancy from the same gene. So, because, as you know, it rains a lot here, especially at harvest time, unfortunately, the seeds that are on the plant can actually germinate, just like you see here, before the plants are harvested. And of course, this completely destroys the quality of the wheat kernel for anything that is usable for human consumption. Although you may find in some specialty food stores sprouted wheat that you can buy, sprouted wheat flour that you can buy, that it will make products, but they'll tend to be very dense and have a sweetish flavor to them because the starch has been broken down into sugars. We've been working on mapping the genes controlling this trait for many years, and so we're now trying to transfer the genes for seed dormancy from an old Kansas landrace wheat variety into our best varieties. 2 of the markers we're using are shown here. Our best marker is BARC55, and this just shows the segregation of BARC55 in a backcross 1 population. Now, remember when I mentioned when you're making a backcross, what you're going to select are the heterozygotes, because you're backcrossing the allele from your source parent into your recurrent parent. So you're segregating homozygous for the undesirable allele and heterozygous with both alleles. And that's shown right here. This is homozygous for the recurrent parent allele, but then you see some that are heterozygous here, and this allele here would be the one that you're most interested in because it's the one carrying the resistance for sprouting. Okay, now I'm going to talk a little bit about disease resistance. One of the most useful aspects of molecular markers is that you can use different markers and you can see them in backcross populations, whereas when you're trying to screen for different diseases, oftentimes if you infect with one disease, you can't see the effect of a second or third disease. This is an example of a marker that it was developed for leaf rust in wheat. We don't have a particular problem with leaf rust in this region, but I think it's a good example of a marker that has proven to be quite useful for some of the breeders out west. They introduced a gene from a wild wheat called Triticum spaltoides that has a 282 base pair band that is associated with a segment that carries the resistance for the leaf rust. So that specific fragment then is selected in the backcross generations, but in order to see the specific resistance fragment, they have to do a digest to separate it from the fragments that are identified in the other genomes. This is what the wheat leaf looks like without LR47. This is a leaf rust resistance that you see from LR47 resistance, and it's a kind of a hypersensitive reaction. The most important disease in our region, at least currently, is a disease caused by Fusarium graminearum, and it causes a disease called Fusarium head blight. This disease is very difficult to screen in the field, and the reason is because the infection only occurs or mostly occurs right at flowering time and within a week or so afterward. In order to get good infections, reliable infections, and a high rate of infection, we have to provide a mist system that increases the humidity in the trial. We also have to spray inoculate with conidial suspensions in order to be sure that every single plant gets infected in this trial and gets infected uniformly. So it's a great deal of labor, and we really need to use molecular markers to be able to follow the genes. Several years ago, a former student of mine mapped the most important gene for resistance to this disease on chromosome 3 in wheat. He lined the QTL from two different populations, and this just shows the LOD plots for those resistance regions. It came from a Chinese variety called Sumai 3. And Sumai 3 continues to be the best source of resistance. They've now identified two other regions in that genome of that variety that are also giving resistance to Fusarium head blight. So one of the markers is GWM 533. The other is BARC 102 that we use. And again, you can see in the backcross generations, we're going to select these heterozygous plants to make our next backcross. So the idea is then to use the flanking markers to identify the heterozygous, then we backcross those into our recurrent parent. And this just shows one of the susceptible and one of the resistant lines. I know it's kind of hard to see in the slide, but it causes the bleaching out and the death of the spikes when they're infected in a susceptible plant. This is Dr. Gary Bergstrom, who is our collaborator, and this is my research support specialist, David Bencher. Another disease that we've just started to work on, we know that it's common in New York, but we've never had the resources to select for resistance. It's called eye spot, and eye spot infects in the crown and the stem just above the crown of the wheat plant, and it causes the death of the whole comb. There are two different genes that confer resistance to eye spot, one from a wild species and one from a cultivated variety. And again, this is another example of selecting in the backcross one generation. We're looking for the heterozygous plants to make the next backcross. One of the viruses we work on is called barley yellow dwarf virus. Barley yellow dwarf virus is transmitted by an aphid, and it infects the plant in the seedling stage. The virus replicates and causes stunting and yellowing of the plants, and oftentimes sterility. One of the sources of resistance is a wild wheat variety called Triticum intermedium. And there's a line that has a translocation on chromosome 7, and the resistance gene is believed to be located in the terminal region of that translocation. We have several markers that we can use, but the one that works the best for us is GWM37. However, this wild segment is transmitted at a lower frequency than a normal chromosome, so we often don't get very many desirable segregates in our populations. Wheat spindle streak mosaic virus is a virus that is transmitted by a soil fungus. It also infects the wheat plants in the fall and is replicated in the plant during the fall and early spring. It causes this chlorotic development of the leaves, and it also causes stunting and up to 30% yield loss. It's very common in this region because it's favored by cool, wet conditions. We have a resistant cultivar called Geneva that we crossed to other varieties, and we're using molecular markers to transfer resistance from Geneva into our susceptible varieties. Now, some examples of insect resistance are shown here. These are primarily a problem in other parts of the country. Hessian fly is an insect that we can find here, but is not very common because most farmers plant late enough in the fall that the fly is not active, so it doesn't lay eggs. It isn't active, and so it doesn't lay eggs before the plants early enough before the plants develop and go into the winter. Russian wheat aphid is primarily a problem out west, and green bug is more of a problem in the south and west. Now I'd like to talk a little bit more about where we think that marker-assisted breeding is going in the next few years. In a breeding program, as you know, our currency are the different alleles for the traits that we're interested in. Alleles have different values, and we need to characterize the value of different alleles. And this requires a great deal of time and effort, partly coming from our earlier work with the molecular markers in marker-assisted breeding. But our primary goals are to validate the quality of an allele, find new alleles, and then use that information in progeny and parental selection. But there are a number of issues. One is that most of the traits in a breeding program are complex. That means that the traits are controlled by multiple genes, multiple loci, and have multiple alleles with unknown value. And breeding programs are very dynamic. That means we're bringing in new genetic material every year. We're discarding old genetic material every year. And so there's a constant turnover of alleles and loci and traits. Because of that, there is always the possibility that we will have markers that we don't know if they're linked to a desirable QTL allele. So we have to be able to reevaluate each and every year for many of the alleles that we see in these segregating populations. Some crops have a problem with low polymorphism, and unfortunately wheat is one of them. And we have to use special statistical treatments to be sure that we're not selecting the wrong alleles, that we know the value of the alleles we're selecting, and that we're not getting false positive readings on some of those alleles. This diagram just illustrates how you can implement allelic value, implement association analysis in a breeding program. All breeding programs start with germplasm. Germplasm that has the characteristics that you're interested in using to improve the crops you're working on. We typically hybridize those different accessions, those different genotypes, to try to combine complementary traits from different sources. That creates segregating populations in which we do selection. When we do our selection, we're selecting for those traits that are important to meeting our breeding objectives. And through selection, we generate new synthetics, new lines, and new varieties. From those, we select individuals that are then tested in replicated trials in different regions and environments. different environments and over years. And that information is then summarized to develop the information we need to identify the best varieties with the appropriate complement of traits that are potential releases to the seed industry and the farmer. If we just add information on the genotypes for the traits we're interested in, for the QTLs we're interested in, we can then use that in association analysis to characterize the allelic value and validate the QTL marker associations that we had earlier or are developing from the new germplasm. That information then feeds back into the selection program through marker-assisted selection or through the parental selection program by identifying those parents that have complementary alleles at the different loci that we've identified. Now I've put this slide in here just to illustrate how markers can be associated with QTL of different effects. This is a diagram that shows kernel length for the three molecular markers. These three molecular markers are shown across the top, and these are just different sized fragments that you would see on those gels like the one that I showed you earlier. Now, what we found was that some fragment sizes are associated with very, very little change in kernel size. Here's one with a slight increase in kernel size. But some are associated with a great reduction in kernel length. What that tells you is that when you have multiple alleles, they have different values. And as a breeder, we want to be sure we're selecting the proper allele. So when you look at those gels and see different fragment sizes, you have to think that I'm actually looking at the trait in a plant out in the field and thinking, well, this plant has a better value for this allele. So here we're looking at kernel length, and some of these fragments are associated with reduced kernel length, which is undesirable. Over here we have two more markers for kernel length. One has an undesirable negative effect, one has a desirable positive effect. And so I want you to understand how different alleles are associated with different QTL alleles of different value. And I'm just going to, yes? You haven't talked about quantitative trait loci in this class yet? Quantitative trait loci are just loci that affect the quantitative traits. So quantitative traits, as you've probably realized, are traits that are affected by multiple loci. And when we refer to quantitative trait loci, it means that we've characterized some of the loci that affect that trait. So we treat those just like essentially major genes because we've Mendelized, to use an expression, we've Mendelized a quantitative trait so that we can follow individual loci that affect that trait. And each of those loci have multiple alleles, just like the major genes. And I mentioned a couple of examples there where we had leaf rust resistance that was controlled by a particular locus that had a resistant allele and a susceptible allele. And they're just different forms of the gene at that locus. Quantitative trait loci are very similar. They have different alleles that are affecting a particular trait. And what we're doing is using molecular markers to be able to identify those different alleles. So we treat them essentially like different alleles for the trait, as long as they're closely linked, and especially if you have good flanking markers. Does that help? Okay. Now I just wanted to wrap up with an example of a recent variety that we released. And I'm giving you this example because I think it's important to understand how complex the genetics are for a variety and how difficult it can be to combine all the complementary characteristics. And remember that in this region we have relatively few difficulties with diseases, and our quality characteristics are not terribly stringent, and we have very few insect problems. In this variety, this was a cross between a variety called Susquehanna and a variety called Harris that I made back in 1988. We just released it this year. So it takes a while. You have to be patient. You have to think ahead. You have to try to combine characteristics that you think are going to be useful 10, 20 years from now. And I developed lines from this cross, which was the 46th cross I made in 1988, and 8138 was one of the selections. We tested this over 5 years in 4 locations per year, and so we characterized the grain yield. Test weight is another trait that is important, because when the farmer takes his grain to the elevator, they do a test weight. And test weight is just simply the weight of the grain per unit volume. And at the elevator, they use quarts. We use liters, but it's the same trait. It's really a measure of how well the grain packs into a container. And that packing is affected by a number of characteristics, mainly extraneous material, broken kernels, sprouted kernels. Those tend to reduce test weight. So it's kind of a measure of overall quality of the grain. We also test lodging resistance, and lodging is that trait when the plant falls over, and it can fall over for many reasons: diseases, high winds, weak straw. And so we use that as an important characteristic to determine if it's acceptable to the farmer, because, as you know, they harvest with harvesters, and it's difficult to lift the grain off the ground if it's lodged. It has resistance to Fusarium head blight, which I mentioned, resistance to wheat spindle streak mosaic virus, resistance to powdery mildew, which I didn't talk about, and then another soil-borne virus, which it's susceptible to. It has good milling and baking properties, which means that it's consistent with the qualities needed for a soft wheat, and it's resistant to pre-harvest sprouting. It's just under 1 meter tall and comparable to other wheat varieties. It's awnless, has white chaff color, and its heading date and its maturity date is similar to other characteristics. Now remember that these characteristics all segregate in every population from every cross that you make. So when you make a cross, you're reshuffling all the genes that are different between those two parents. Most of the traits that you're interested in are quantitative. That means they're controlled by multiple loci. So you can imagine that there are dozens, if not hundreds or thousands of genes that are segregating, and it's a plant breeder's job to sort out those desirable segregates that can be potentially released as varieties. This is just to acknowledge some of the people on our project. Emily is one of those people. We have several graduate students on our project, and postdocs. James Tanaka and David Bencher and Gretchen Salm are our technical assistants. We get support from the USDA for our work, and we work closely with the Grain Genes database. Okay, I guess I'll stop there and try to answer some of your questions.
So how many traits do you select for a certain time?
We are pretty opportunistic about selecting segregates for different traits. So for example, some years we really don't get very much powdery mildew, so we can't very well select for that trait. Other years we get a lot of powdery mildew, so we then, in that year, we do very intense selection. Typically, I only like to look at one trait at a time. That allows me to train my eye and focus very carefully to be sure that I evaluate each line the same way. Heading date is typically done at one time. We try to pick about a mean of the flowering time for all the varieties, walk through and make notes on when some of them flower, and when the ones that are too late, we discard those. Fusarium head blight can't be scored in a regular breeding nursery. It has to be scored in a special nursery where we've inoculated with a pathogen and where we've provided additional moisture. So that is done at a very carefully prescribed time that occurs when we see the maximum difference among the varieties in a trial. It doesn't do any good to go out and score lodging, the lodging trait, when there's no lodging or when they're all flat. If they're all flat on the ground, some of them may be lodging resistant, but you won't know that. So the idea for most traits is to maximize the variation due to genotype and minimize the variation due to the environment. Some traits you can score, some you can't. Yeah, that help?